Okay, so this lesson is going to take you through the work of Pavlov and all his experiments that culminate together in his salivation of dogs and is the principle behind classical conditioning. So one of the things that Pavlov discovered almost by accident really was the way in which the dogs salivated initially naturally to food being brought in by a lab assistant, but further down the line he found that they began to salivate when the lab assistant was present without food. This gave him the background ideas to start to consider why it was that they'd learnt to behave in that particular way. So back in the 1920s, he conducted his research with the dogs. And in 1927, he gave what we call a series of lectures. So this was at local universities about all of his experiments. He conducted approximately 37 in total, but these are drawn together in sort of one summary of all the work that he'd actually done. So all of those experiments come together in what we understand now as Pavlov's research with salivating dogs. So what I would like you to do, first of all, just to set the scene and give yourself a little bit of background knowledge, just spend about 10 minutes having a look at Pavlov in more detail. This will give you an idea about what he did, what his studies were about. Make notes on a piece of paper, put that in with your learning notes folder at this particular point. Don't worry too much about going into his studies in detail, just the general ideas, what he found, what he discovered, how he went about looking at salivation. Might be helpful to start considering animal ethics. So how did he collect the saliva to measure sort of how much the dogs are salivating and things like that. So pause the lesson for about 10 minutes. Have a look, make some brief notes or a mind map, and then restart, and we'll go through Pavlov in a much more detail. So Pavlov was technically a biologist, and he was interested in looking at how the cerebral cortex worked, and he was focusing really on animal behaviour at the time. What happened with the salivating dogs was that he wanted to look at natural associations between stimulus and inherent reflex responses. So those reflex responses are things like your blink reflex. The reflexes, you know, if you put your hand on something hot, your arm moves away immediately. They are reflex responses that you don't physically control yourself. You don't need to worry about them. They just take place automatically. And Pavlov was wondering sort of what it was within the cerebral cortex that actually created those associations to trigger those particular reflex responses. The purpose of using dogs rather than any other animal is essentially they have what we call higher order thinking skills. So dogs are an animal that can be trained quite easily. They can be led into understanding associations. You can teach a dog to sit, although I can't teach Daisy to sit. She ignores me, but apparently you can teach dogs to sit, fetch, run, roll over, all of those different things. So whilst they don't understand our actual language, there is some element of higher order thinking skills that dogs can employ in order to be able to make those kinds of associations. In addition, they're quite manageable in terms of housing and care. So when undertaking scientific tests, it's not difficult to appropriately house and care for a dog undertaking scientific testing. It's possibly quite useful if you want to do a little bit of independent reading around the subject. There's some stuff on Teams, there's a few videos around this. Lots and lots of learning theory that we're going to have a look at use animal research. And one of the key things in our methods for this section is to actually consider animal research and animal ethics. We do so by looking at Scientific Procedures Act and the Home Office regulations around animal research. So it would be handy if you want to start having a look at those now. They will be coming into some of your studies as evaluation points. Obviously, you can't use human ethics. We don't expect animals to be able to do daft things like consent because they can't. but there are a wide range of very specific animal ethics surrounded by animal research. So it would be really helpful to start looking into that and start building a little bit of wider reading around the Scientific Procedures Act and Home Office regulations. Another aspect that we begin to pick up through learning theory is what we call determinism. Now, this is a wider concept across psychology that kind of looks at how predetermined your behaviour is compared to how much free will and choice you have over those particular behaviours. It's the kind of thing that is focused on in biological psychology and learning overall. This idea of a predisposition or something that you have no control over. So Pavlov talks about the fact that responses such as fear and anger, those reflex, innate biological responses that tend to come from evolutionary traits, are reflexes that are stimulated by the cerebral cortex and therefore result in what we call observable behaviours. Now think back to the introduction to learning theory. Remember, they are only interested in stuff they can see. Behaviourism or learning theory is about what can you observe. It's all about empiricism. So if you can't see the outcome of a behaviour, it is not something that they would look at in terms of research. Now, like biology, this aspect of learning theories is very deterministic. Particularly Pavlov's research with animals, but across learning theory as we go through the rest. They believe that things like those reflexes are inbuilt in your brain. They occur in response to certain stimuli in all humans or breeds of animals. They are a universal, inherent, predetermined trait, which is why it is seen as deterministic, right? So if you think about things like your blink reflex, if something comes too close to your eye, you automatically blink. You don't control it. You can't actually stop that happening if something is sort of heading towards you that could cause damage to your eyes or your optical nerves. So Pavlov argued that things like that are determined by your cerebral cortex, and that actually all people, unless of course they have some form of brain dysfunction, would show that reflex behaviour. Every single human being and the vast majority of animals will respond with a blink reflex should something come at speed towards their eyes, and you have literally no control over that. So that's where they're coming from when they talk about this idea of determinism. You are not making an active choice. You are not choosing to behave or respond in this particular way. So having done a little bit of background reading into Pavlov, it's obvious that his main focus was the trigger of salivation in dogs at unexpected times. So what had happened, he was doing cerebral cortex investigations on the animals, and he began to start his work into what became classical conditioning when he noticed that the salivation in the dogs began at times that actually surprised him. Times that there was no food around, that they shouldn't actually be salivating because there was no stimulation for that. So stimulation for salivation comes in a couple of different ways. The sight and smell of food can result in salivation in dogs. That's a natural reflex. It should occur, and it should also predominantly occur when the food is in the mouth. That's when most salivation is triggered, because it is a reflex that aids digestion. It serves no other purpose other than to help break down food in the digestive tracts as it kind of goes down into your stomach. And it is the same for dogs as it is for humans. So what happened was that the dogs were beginning to salivate at times when that food was not present, you couldn't see it and you couldn't smell it. So it became quite clear to Pavlov that actually the reflexes were associated with something else. They weren't working alone. It wasn't that, the food hit the mouth and it started the biological process. Something else was going on. Now he put this down to cerebral cortex functioning, so a connection between the stimulus as it was and the reflex of salivation itself. And what he began to consider was how reflexes were actually linked to learning mechanisms. How did you learn particular behaviours and responses that actually shouldn't take place at specific times? So in the case of the dogs, what had happened was the guy that used to basically feed them came in with his white lab coat, all the rest of it, and fed the dogs. And what they did was they associated food with the lab assistant. So whenever that lab assistant became present, was in the lab without food, the dogs began to salivate because they had an expectation. They were prepared, and all of a sudden that natural trigger or that natural stimulus of food became attached to an unnatural stimulus. So if you think back to the work on classical conditioning, you've got your unconditioned stimulus of food, your unconditioned response of salivation, and you've got your attachment to a neutral stimulus, which is the guy in the lab coat. Once that attachment had been formed, so the association and pairing took place, the guy in the lab coat became your conditioned stimulus, and the conditioned response was the salivation from the dogs. So after he'd noticed this process, Pavlov wanted to see if he could actually create an association or pairing between a neutral stimulus and an unconditioned stimulus by himself. So could you actually create this conditioned response to whatever it was that you wanted to? So Pavlov used the dogs and he presented them with the sound of a metronome and a bell. So it depends on which study he was doing. In terms of your exam answers, you can use metronome and you can use a bell. It doesn't matter which phrase you use, they're both interchangeable. So he wanted to see if the dogs would become conditioned to salivate to the sound of a bell. So the UCR, your unconditioned response, was what he wanted to associate to a conditioned stimulus of a particular sound. So as he went through, it is referred to as meat powder. For anyone who has a dog, if you feed them dry food, that's what meat powder was back in 1927. So that's what he means when he's talking about powder. But anyway, it was presented at the same time as a ticking metronome or the sound of a bell, with repeated pairings to create the conditioned results. Okay, so this is quite a nice way to express the stages in a Pavlov's procedure. So what he did was he started off with the first test of the dogs. So this is before any conditioning takes place to any kind of neutral stimulus. What Pavlov did was he tested whether an unconditioned stimulus, which was in fact the food, actually did trigger the response that he was after, which is the unconditioned response of salivation, all right, in the dogs. Once he had checked that that was a naturally occurring response that was already in place, Pavlov then moved on to checking the neutral stimulus, and this is really important. You'll come across checks like this in Pavlov, and again when we go back to Watson and Rayner, where they conditioned little Albert. So Pavlov presented to the dogs what he thought would be a neutral stimulus. All right, so that's the bell. He presented it to see what the dogs actually did, because actually if they salivated or behaved in a particular way, then it wasn't a neutral stimulus. So he needed to check that there was no association to the sound of a bell or a metronome. So he checked their response and nothing, right? The dogs didn't salivate. So he knew that the neutral stimulus in his experiment did not cause any form of salivation response in those dogs. So he moved on then to stage 3 of the procedure. And what he did at that point was he presented the food which he knew would produce salivation. And at the same time as presenting the food, he made the noise with the bell or the metronome. So the two, all right, were paired. All right, this association where they were added together, the pairing process began. Right now, the response from the dogs during this stage of the conditioning process is still considered an unconditioned response, because actually for the first few steps of this, they're responding to the food. All right. As this occurs more and more and more often, that's when the association between food and bell begins to form. All right. So once he was sure that he'd presented the bell and the food at the same time, often enough, all right, with the salivation response, he decided to withdraw the food and to see what happened basically after the conditioning process. So what he did at this point was he would present the sound of the bell and measure the response of the dog. Now, where the dogs salivated, right, that indicated that the bell had become the conditioned stimulus. So it had changed from its position as neutral stimulus, all right, before conditioning and through the pairing process. It had changed to a conditioned stimulus because it generated the response of salivation, which became a conditioned response. It was a salivation response that had been conditioned or associated to the conditioned stimulus. So this was the stage at the end, right, after condition 4, that Pavlov was able to determine that you can condition responses, natural reflex responses that really shouldn't occur in these situations, to neutral stimuli. Right, so the neutral stimulus became the conditioned stimulus, right, and the unconditioned response, the unconditioned response of salivation became the conditioned response. Okay, so you've got a small study sheet for Pavlov. This is not a classic study for this section. It is not one that you need to know in huge volumes of detail. It's more of an overview of his research overall, a bit like Milgram's variations and things like that. So you don't need to do a full A3 sheet. If you want to, you can, by all means, but really the only sort of depth you need for this is an A4 sheet, and it would come up only ever in the form of an 8-mark question, only on paper 1, and it wouldn't be assessed anywhere else. So what I'm going to do is go through some more specifics and the key features of the procedure. You can copy those down onto the A4 sheet and just make a note of the steps and stages, being able to identify things like the IV, DV and key steps in the procedure. So you might want to pause at the moment, grab that study sheet and just get ready to make some notes. Okay, so Pavlov's aim was to find out if a reflexive behaviour, so that was your salivation, can be produced in new situations through learning. In particular, he was focusing on the reflex to a neutral stimulus to see whether learning can take place to produce a conditioned reflex in new situations. So that was his official aim. In terms of design, it's a repeated measures design because the dogs were in the pre-conditioning, conditioning and post-conditioning assessments. So each participant, being an animal in this case, repeated the various stages of the procedure. It's also possible to refer to it as a within participant design because it's the same individual that, or animal in this case, that goes on through each individual process and step in the procedure. Whichever way you refer to that is absolutely fine. So the IV for Pavlov is the natural reflexive behaviour, salivating when food is in their mouth. The other condition of that test is after they've been conditioned. So have they associated the food with a different or previously neutral stimulus? So it is a case of the impact on salivation of a unconditioned stimulus and compared to a conditioned stimulus, a previously neutral stimulus. The dependent variable was whether they salivated. Now, if you did find this out in your research, what he did was he measured the number of drops of saliva the dogs produced. He did so by an incision in the dog's throat or jowl, just towards the back where the saliva glands are, and inserted a tube that dripped into a little canister so that he could physically measure and empirically see salivation and test whether it was, comparisons of things like, did they salivate as much to the bell as they do to the actual food, and those kinds of checks that he could have a look at. So that is how he empirically measured his dependent variable. The sample consisted overall of 35 dogs. He used a variety of different dog breeds, so he didn't just pick one type of dog. So in terms of generalizability or representativeness of a broader range of dogs, it's quite generalizable. Whether that would be the case now, if you're doing evaluations, we now have hundreds of dog breeds. Back in the 1920s, that wasn't really the case, but he did select enough to make this representative of a wide range of dogs. They were also raised in the kennels in the lab, and that's really important because this was their home and this was their natural environment, what it means is that actually it doesn't lack ecological validity for the animal, because this is where they are used to, this is what they're used to being in. So whilst we normally talk about artificiality and whether or not the dogs would behave in this particular way in their own environment, the reality is this was their environment. Now, it doesn't represent the environment of all dogs, because most of them, if you can hear mine barking in the garden, live in a house or a home and that kind of thing. But for the particular dogs he tested, they were lab raised in the kennels. So for the procedure, Pavlov placed each dog in a sealed room that didn't allow the dog to see, smell or hear anything outside. This was really important to control and prevent other stimuli or extraneous variables from making the dog salivate. It is something that's extremely important when you're doing conditioning, to make sure that it is silent and there is no other stimulus available is essential, primarily because you don't want that association to form with a confounding variable that you hadn't planned for. So it has to be really, really well controlled to make sure that those variables don't skew your data. The dog was strapped into a harness to stop it moving about, and its mouth was linked into a tube that drained saliva away into the measuring bottle. So I'm going to show you a diagram of what that looks like in a second. Ultimately, the harness to prevent movement and things like that, that's where you would start to bring in here sort of a bit of task validity. No matter how much they were raised in a kennel in a lab, being strapped into a harness was not their normal kind of activity. So you've got the task validity activity sort of evaluation in there. So just remember that when you're looking at either ecological or task validity for the participants in Pavlov's research. So you can pause now for a second if you need to copy any of that onto your sheet, and then restart, and I'll just quickly talk you through the diagram of how the dogs were strapped in. Okay, so this is a rough diagram of Pavlov's experiment. It's done quite nice and kindly here. It's not quite, if you have a look at that, how the saliva was collected. In actual fact, the collection tube was more likely to be around here, where the back of the saliva glands are, and it was cut into the throat. I think that's important for you to just kind of make a note of that. Although it's not completely unethical, there is no evidence in his notes that he gave them an anaesthetic when he did that. So you could talk a little bit about unnecessary harm if you wanted to. So this was how a dog was harnessed in. The collar, forelegs and hind legs were put into a harness along with their paws at the bottom, right? So this was to restrict their movement, so that the salivary gland tube didn't fall out on a practical level. B, dogs get very, very excited when you present them with food. They jump around, and it makes things quite difficult to test. In addition, because the tube was actually inserted into their jowl and their saliva glands, had they been bouncing about and jumping around like that, they could have caused themselves damage and harm as well. So, you know, it wasn't completely bad to harness them in. However, it does reduce the natural position that a dog might be in for feeding. So the dog was strapped into this harness, right, that kept them in place. They were in a silent room. Obviously, you can't see that because of the diagram, but basically this was a box here. All right, so that would have been a wall, and then another one at the back with a roof, and it had a little window here where the researcher, there we go, let's put the researcher in, brought the food to give to the dog. All right, so as that took place, the tubing, right, was coming to the outside of the room so that they didn't have to interfere and become any sort of extraneous variable. It was collected in a tube, passed down, and then a recording of the number of drops was made. So there was nothing at all influencing those dogs other than the presentation of the food and the sound of the bell at the same time. You will not get asked to draw this diagram or, you know, anything in any great detail like that on your exams. But I think it just does help put it into perspective as to sort of how they were harnessed in to help you do those evaluations. So Pavlov had a baseline control condition. So he presented the dog with food through the hatch and the dog salivated. Right? That was necessary in order to measure the difference in saliva to what would be the experimental conditions. And the experimental condition was when the food was presented with the neutral stimulus, which was the bell or the metronome, at the same time as the food. So you had those various stages that we saw on the diagram earlier. To condition the dog, he paired the sound with the presentation of food approximately 20 times. Now, it wasn't always 20 times. It depended on how attentive the dog was. So if you had quite a keen dog with, you know, higher order thinking skills, quite happy to pay attention to humans, it might have been slightly less. If you had a dog like mine who pays absolutely zero attention to humans ever, it'd have probably taken about 100 goes to get her to do what she was told. So it did depend on the attentive nature of the dog, but it was approximately 20 times overall. After the conditioning process, so that association between the bell and the food, Pavlov presented the dog with just the sound, so just the bell, no food, and he measured then the salivation that happened. So results of Pavlov, he found that the conditioned dog started to salivate 9 seconds after hearing the sound, and by about 45 seconds had produced a total of 11 drops of saliva. Again, their average, he used 35 dogs, is the average results. You do not need all 35 dogs in sort of individual depth. You just need to kind of know the overall findings. Took about 9 seconds, after the bell went off, and about 45 seconds in, 11 drops of saliva had been produced. That would be the measure of whether or not the neutral stimulus had become a conditioned stimulus. So his conclusions overall led to his theory of classical conditioning, and it's from there that further research has taken place, attachments to humans have taken place, and obviously the therapies and treatments for phobias that we're going to have a look at further on in the course. So he concluded that a neutral stimulus, after being repeatedly paired with an unconditioned stimulus, can turn into a conditioned stimulus, producing what is now termed a conditioned response, because it has been unnaturally associated with a different stimulus to what it should actually be associated with on a normal level. So Pavlov explained that that was how the brain learned to see the new sound as some sort of signal. The cerebral cortex made the links to the reflex response, and therefore those new conditioned responses could be formed. He applied this really to how animals in the wild would learn to hunt or escape being hunted, because they're actually able to learn to apply their reflexes to new situations based on experiences they've had before. So it explains why animals would be scared of humans, for example, where in actual fact the human might be bigger or less physically able to, challenge the animal, probably get eaten by some animals, but actually they've associated the humans, the hunting, shotguns, all those awful things, and they've learnt that this is not a creature you go near, no matter what. So it's things like that that Pavlov was interested in. Remember, he was originally a biologist looking at these kinds of things. He began his life actually as a vet, so that's where he had a fascination with animals from. So all of his research applied to the ways in which animals could learn behaviour, and once he'd created classical conditioning, people began to test this in relation to human beings, because at the end of the day we are in fact animals like all other animals on the planet. So you might want to pause again, finish copying up that bit, the procedure, results and conclusions, and then once you've got all that written down, just restart the lesson. Okay, so a quick recap activity for you, just to make sure that you know how you're applying your research methods in this particular context. Pavlov obviously did lab experiments, which we learnt about in cognitive psychology. So you have got 60 seconds to very quickly describe the key features of a laboratory method. Get down as many of the key points as to what makes something a laboratory experimental method. And your 60 seconds starts now. Okay, that's 60 seconds up. Ironically, having used that alarm for all your assessments over the year, you are all too well conditioned into the sound of that. That will instill fear probably for the remainder of eternity. So hopefully you've got down some key points. If you sat with someone, you can chop and change, share your ideas. Ideally, you should have artificiality of the setup, control of extraneous variables, independent variable, a baseline condition, an empirically measurable dependent variable. Some of those are key features. Okay, so the last little bit of this lesson, going into homework if you need to finish it, you should have plenty of time to work through these, is to think about the application of your Graves evaluation points to Pavlov's study. So I want you to come up with strengths and weaknesses. Same as everything else, your A1 is the strength of the study or the weakness of the study that you find relevant. Remember, generic answers score 0 marks. You can't just talk about being a lab. You can't just talk about it being an animal. You have got to be really specific, picking those bits out of Pavlov. And then your AO3 link to Graves and how or why that makes it a strength or weakness. You know, remember to tell the examiner that I think this is a strength because. If you don't give the because, you're not going to get those AO3 marks in there. Example that you've got on screen, so none of you can have this as your own examples. So, you know, you can put this on your notes, but you've got to come up with more than this. Pavlov used a repeated measures design where the same dogs were tested in both conditions of the IV before and after the pairing. All right. This increased the internal validity of the study as Pavlov can be more certain it was the effect of the IV pairing of food in the bell that resulted in the conditioned response of salivation as the DV and not other differences from the nature or cognitive abilities of the dogs. Right, so consistently throughout that answer, tying it back in to dogs, salivation, food, bells, pairing, and so on. So they're the kind of things that you need to do to tie those together. So I do know it says on screen, and how we been in lesson, this is what would happen, that you're going to be put into 2 groups. I'm not going to put you into 2 groups for that, but I do think you should buddy up with 2 or 3, maybe 4 other people. Each do 1 strength, dish them out, so strengths, weaknesses, do 1 each, bring them together and share them, reduce your workload, all right, pull all your stuff together. Don't sit there and try and do 4 strengths and 4 weaknesses all by yourself. Work with other people, allocate 1 each, bring them all together and share them between you, all right. So that's what I want you to do for the remainder of the lesson. Homework, which needs submitting on Teams to this assignment, if you have a look at the next slide, is your straightforward 8-mark essay, evaluate Pavlov's studies of salivating dogs. Right. So once you've all worked together, pulled together those strengths and weaknesses, had a chat about them, shared them and so on. And if you want to share them on Teams, that might be really helpful. Start a chat in the learning theory section and each put 1 strength or 1 weakness into it. And then the whole class can have, you know, 20 strengths and weaknesses between yourselves. That would be my recommendation. Once you've got all of those, pull them together, do me a standard evaluation of Pavlov's studies, and attach this answer to Teams just so that I can see that it's been done and we've pulled it all together. Keep a hold of it because I do want to take it in on paper and actually annotate and stuff like that when I mark it, but just attach a copy for me because I will start to have a look through these and try and get you some feedback as soon as I can. Got any questions? Post a message on Teams, make sure you tag me in it because otherwise it doesn't ping through on my phone. Drop me an email or ask me in the next lesson. All right, guys, thank you very much.